Good afternoon to all the NEET aspirants. This is your Prasanna Ma'am from NEET World, and here we are going to discuss the Advanced Questionnaire from Biological Classification. So today there is something interesting that we are going to exercise the Advanced Questionnaire with concept revision, concept by concept from the chapter Biological Classification. So let's get started with the first question, ma. So here in this question, the question is from 5 kingdom system of classification. So in 5 kingdom system of classification, which kingdom out of the following can include blue-green algae, nitrogen fixing bacteria and methanogenic archaebacteria? So yes, we have 5 kingdoms, right, in 5 kingdom system of classification, which is given by R.H. Whittaker, We have 5 kingdoms. What are they, ma? First is Monera, second will be Protista, third fungi, fourth Plantae and fifth is Animalia. So monerans include all the unicellular prokaryotic organisms, yes or no, ma? So monerans include all unicellular prokaryotic organisms. Protistans include all the unicellular eukaryotic organisms. So this includes all the unicellular eukaryotic organisms. So what is the difference between prokaryotic and eukaryotic here? Here these cells will not have membrane bound organelles, they do not have well defined nucleus, they do not have a membrane bound nucleus. Whereas in the unicellular eukaryotes, they have membrane bound organelles within the cell and they have well defined membrane bound nucleus. And in fungi, if you see, all are heterotrophic, multicellular heterotrophic organisms. Here also organ type, okay, autotrophic organisms, okay, multicellular autotrophic organisms, multicellular heterotrophic organisms, all these types of organisms are included in the respective kingdoms. So when we have to focus on the Monera and Protista, monerals include blue-green algae, which is nothing but what? Cyanobacteria. Cyanobacteria is a prokaryote, yes or no? Yes. Cyanobacteria is a prokaryote which is a blue-green algae. So this will be helpful in the nitrogen fixation, especially in the paddy fields. Some examples of cyanobacteria or blue-green algae include Oscillatoria, Anabina, Alocera and Nostoc. So they come under kingdom Monera only, and methanogenic bacteria or archaebacteria, eubacteria all comes under Monera, kingdom Monera only, because they are prokaryotic cells. Whereas Protista includes 3 different types of unicellular eukaryotes. One are plant-like, best example is chrysophytes, dinoflagellates and euglenoids. Second type is fungi-like unicellular organisms which are slime molds, and animal-like are protozoans. Animal-like are what? Protozoans. We have amoeboid protozoans, sporozoans, flagellated protozoans and ciliated protozoans. As such, we have what? Many different types of unicellular eukaryotic and prokaryotic organisms. Blue-green algae, which is cyanobacteria, which is also nitrogen fixing bacteria, and then methanogenic archaebacteria, they come under what? Monera. Under Protista, all unicellular eukaryotes are included, plant-like, fungi-like, animal-like, which include chrysophytes, dinoflagellates, euglenoids, slime molds and protozoans. And under Plantae, all the multicellular autotrophic organisms, and fungi, this is multicellular heterotrophic organisms. So here option A is the right answer. And then if we have to look into the next question, ma, which is match the following type of question. Mostly from biological classification, if we have to look into previous year papers, example based questions are only more. So highlight the examples, give additional interest on the examples when you are going through this chapter. So here if you see, match column 1 and column 2, choose the answer from the codes given. Escherichia coli. E. coli is what? Rod shaped bacteria. Okay, E. coli is rod shaped bacteria. And Streptococcus is spherical shaped. Streptococcus is spherical shaped. Treponema pallidum is helically coiled. Treponema pallidum is helically coiled. As such, what do we have? E. coli, which is rod shaped. 1 is 3 and 2 is 1, then 3 will be 2. Option B is the right answer. Okay. And then, yeah, here again the question is about archaea and 5 kingdom system of classification only. Look into it keenly. The domain archaea is not characterized by, okay, which of the following is not the characteristic for archaebacteria or the domain archaebacteria. So, who introduced these domains? Carl Woese has introduced 3 domains. Carl Woese has introduced 3 domains and 6 kingdom system of classification. 3 domains include what? Archaea, bacteria and eukarya. Under archaea, archaebacteria are included. Archaebacteria. Under bacteria, monerans or eubacteria are included. Eubacteria. And eukarya includes 4 kingdoms. Those are protista, fungi, plantae and animalia. Clear, right? So, this is about 3 domains and 6 kingdom system of classification. So, the domain archaea is not characterized by pseudopeptidoglycan in cell wall, pseudopeptidoglycan is present in archaea which has lipopolysaccharides in its cell walls, okay, whereas true peptidoglycan is observed in the cell wall of eubacteria. Note down that difference, primary difference in the cell wall between archaebacteria and eubacteria. And then linear chromosomes, introns present in some genes. Yes, intron exon arrangement is observed in archaebacteria and then in eukaryotes again, not in bacteria. Methanogenesis as unique metabolism. Yes, methanogenesis or methanogens are observed where? In the archaebacteria itself, methanogens. So, here linear chromosomes are not observed, linear chromosomes are observed only in the eukaryotic cells, whereas in archaebacteria or eubacteria you can see the circular chromosomes itself. Next question, if you have to consider the following regarding the fact that cyanobacteria are now kept in Monera, not in Plantae. They are prokaryotes. Cyanobacteria are now kept in Monera, not in Plantae. For what reasons he is asking? Yes, they are prokaryotes. Cyanobacteria are prokaryotes. Their cell wall of cyanobacteria has peptidoglycan, true. They can fix atmospheric nitrogen, correct. So, all the options 1, 2, 3 are correct only. So, with this question, once let us recall the cyanobacteria structure also. So, cyanobacteria or nostocs, they can be any type, unicellular, colonial, filamentous, but nostoc usually will be the colonial nature or filamentous nature like this. It has 2 cells, one is vegetative cell and the second type will be generative cell. So, vegetative cell or generative cell are covered by what? Gelatinous sheet. Vegetative cell or generative cell are surrounded by the gelatinous sheet. So, it has 2 cells, vegetative cell and one large heterocyst. So, it has vegetative cell and heterocyst. Vegetative cells have chromatophores which take the sunlight, perceive the sunlight and they will conduct the photosynthesis, photoautotropic in nutrition, whereas heterocyst will do what? Nitrogen fixation. Heterocyst will conduct what? Nitrogen fixation. They will fix the nitrogen. So, vegetative cells will conduct what type of photosynthesis? That is the question. Oxygenic photosynthesis where oxygen is evolved, whereas heterocyst walls are very thick, impermeable for gaseous exchange or impervious for the gaseous exchange, wherein this will fix the nitrogen in anoxygenic conditions because of the enzyme nitrogenase. Remember that it is because of the enzyme nitrogenase. Nitrogenase will fix the nitrogen. This nitrogenase will be inactive in the presence of oxygen. That is why the wall layers of heterocysts are thick, which are impervious for the gaseous exchange. This is a small physiological concept in cyanobacteria that you people have to be aware of. So, with respect to this question, they are prokaryotic. Cell wall, as it is eubacteria only, cell wall is made up of peptidoglycan and they can fix the atmospheric nitrogen. Option D will be the right answer. Next question. Consider the following statements regarding mycoplasma. They are the genus of bacteria, yes, yeah. They are the genus of bacteria that have a unique cell wall of their own. Yes, mycoplasma has a genus of bacteria and they have unique cell wall. Wrong, because mycoplasma are wall-less bacteria, or they are popularly known as bacteria with their coats off, okay. And then their genetic material UGA codes for tryptophan. Yes, this is true. None of them are pathogenic to humans. Wrong. Mycoplasma aeruginosa, Mycoplasma pneumoniae, they cause human diseases. So, this statement is wrong. They have a large genome. Wrong. They have a very small genome. Okay. They can be cured by penicillin. No, penicillin will act on the cell wall of bacteria. Cell wall of bacteria, as mycoplasma are wall-less, so it will not act on the mycoplasma. They can survive without oxygen. Yes, they can survive without oxygen. They are called as facultative anaerobes. Facultative anaerobes. Most of them are facultative anaerobes. So, here how many statements are correct right now? So, one that is in the genetic material UGA is scoring for tryptophan, and then they can survive without oxygen. These two statements are correct. Mostly in your examination, multi-statement questions are asked like this only, like a couple of statements are given and we have to identify whether correct set of statements or incorrect set of statements. For that, deep analysis of the concept is required, okay. So, this level of practice is required for your exam, is what I felt after looking into NEET 2025. And then, yeah, so here you look into this question, yep. The given statements describe a group of organisms, okay. So, the given statements will describe group of organisms first. Instead of cell wall, they have a protein rich pellicle making their body flexible. So, which organisms have a protein rich pellicle instead of cell wall? Definitely it is euglena, okay. And then they have two flagella, one short and one long. Yes, they have steichonematic flagella and one of them is thin cell type. They have mixotrophic nutrition. In the presence of sunlight, they are autotrophic. In the absence of sunlight, they act as saprophytic. They conduct saprophytic mode of nutrition. They are the connecting links between plants and animals. Yes, in Protista, when we are discussing about plant like protists, we have chrysophytes, dinoflagellates and euglenoids. Euglenoids are the connecting link between the plant like protists and fungi and animal like protists, just because of their mode of nutrition, which is mixotrophic, okay. So, yes, this is also true. And then, yes, they show mixotrophic nutrition. So, all these features are with respect to what? Euglenoids, that is option D. Clear right now? Next question. Yeah, this one. A slide under a microscope shows the following features, okay. Unicellularity, yes, that will show unicellularity, and then well defined nucleus is observed, true, well defined nucleus. Biflagellate, one long flagella and one transverse flagella. So, unicellularity, well defined nucleus is for unicellularity and well defined nucleus. With this two we can confirm it is a protistan, okay. With this feature, biflagellate, one flagella lying longitudinally and one flagella lying transversely. This feature is only with respect to what? Dinoflagellates. Dinoflagellates. So, this dinoflagellates with the longitudinal flagella and the transverse flagella, they will show the whirling whip movement. Whirling whip movement is popularly, it is only applicable for what? Dinoflagellates, because of the longitudinal flagella and the transverse flagella. So, here what is the organism, what complete features or the set is for what organism? Dinoflagellates, which are plant like protists. Few of the dinoflagellates like Noctiluca, they will show bioluminescence. Few of the dinoflagellates like Goniolax will form red tides in the Red Sea. Okay? And then, yes ma, you will get the PDF of this question in our app NEET World. You can install that from the Play Store. There you can get the PPTs, PDFs of all the questionnaire sessions that we are doing. Okay? So, install that app and then you will get it ma. The link of that app will be provided in the description. Okay? So, here assertion reason type of question, if you see assertion, slime molds form fruiting bodies during unfavorable conditions. Yes, slime molds, they will increase in their area or their plasmodium extensions, they will increase in their feet during favorable conditions. During unfavorable conditions, they will give rise to fruiting bodies. Slime molds will form fruiting bodies during unfavorable conditions. Okay? And then in reason, naked plasmodium is formed during favorable conditions. Yes, naked plasmodium is formed during favorable conditions. Okay? So, both assertion and reason are true, but in reason it is nowhere explaining about the fruiting bodies. These are two different statements and correct statements. In that case, for what option we have to go ma? Option B. Both assertion and reason are true and reason is not the correct explanation of assertion. And then next question. So, this question, look into that. Consider the following statements. The characters not shared by fungi and other eukaryotes. Look into that carefully. Nuclear envelope. Nuclear envelope is present in a fungal cell and other eukaryotic cells also. Membranes with ergosterols. Ergosterols are specific for fungi only. Remember that ma. Specific for fungi only. Okay. So, membranes, cell membranes or plasma membranes with ergosterols, this is specific for fungi only, not for other organisms, not for other eukaryotes. For other eukaryotes, cholesterol will be present. Okay. Other eukaryotes, cholesterol is present. Fine. Cholesterol is present in the cell membrane of the other eukaryotic cells, whereas specifically for fungi, ergosterol is present. ATS ribosomes present in all the eukaryotic cells. Split gene arrangement, that is intron-exon arrangement. Intron-exon arrangement is observed in all the eukaryotic cells and in archaeal cells also. So, here which is not common for fungi and other eukaryotes? Option 2, only 2. Okay. So, A is the right answer over here. Okay. And then next question. Match the following type of question ma. Look into that carefully. Match column 1 and column 2. Select the correct option from the codes given below. Edible delicacies. Edible delicacies we can include morels and truffles. Edible delicacies, morels and truffles, that is option 4. And experimental genetics. Neurospora crassa is the best example for experimental genetics, that is option 2. And source of antibiotics. Source of antibiotics, penicillium and streptomyces. Penicillium notatum, penicillin antibiotic, streptomycin is the antibiotic, that is option 1. And rust and smut fungi includes what? Puccinia and Eustilago. Puccinia, which is the rust fungus, Eustilago is the smut fungus. Both of them will belong to what? Basidiomycetes class of the fungus. Basidiomycetes. All the example based questions are very much important for your exam. From this chapter though, many a times the questions are directly asked from the examples only. Okay. So, here what option will be right ma? A is 4, yes, B will be 2 and C is 1, D will be 3. Okay, D will be 3. Option C is the right answer. And then next question. Yes, this one you observe. Recognize the figure and find the suitable matching. Recognize the figure and find the suitable matching. So, A is what? First you identify the images properly. A is what? Tobacco mosaic virus. Tobacco mosaic virus, where it has single stranded circular RNA and it has protein subunits, that is capsid. Capsid has this protein subunits which are capsomeres. This single stranded circular RNA is having 6400 nucleotides, whereas this capsid, which is the proteinaceous sheet, has proteinaceous subunits called as capsomeres, which are 2130 capsomeres. Okay, this is about tobacco mosaic virus. And here if you see, this is bacteriophage. Bacteriophage, wherein it has a head, neck, collar and tail. Tail has base plate, pins and tail fibers. At the end of this tail fibers, it has an enzyme called as lysozyme. This lysozyme will degrade the cell wall layer of the bacteria, which will help to deposit this genetic material which is present in the bacteriophage directly into the bacterial cell. What type of genetic material is present over here? In bacteriophage, double stranded circular DNA. Remember the genetic material type, very important. So, this is the brief introduction about TMV and bacteriophage. Now, let us look into the statements which are given. First, A attacks on eukaryotic organism while B attacks on the prokaryotic organism. TMV is attacking on the eukaryotic organism only, that is tobacco plant, while B on the prokaryotic cell, that is bacteria. A is correct only. Statement 1 is true only. And then A has single stranded RNA as genetic material, while B has double stranded genetic material, that is DNA. True. A belongs to Protista, wrong. A does not belongs to Protista, that is tobacco mosaic virus, and B belongs to Monera, wrong. Both A and B of them does not belong to any kingdom in Whittaker's 5 kingdom system of classification. I would like to highlight here two things. From Whittaker 5 kingdom system of classification, what are escaped? Viruses, viroids, prions, lichens, these are not included in Whittaker's 5 kingdom system of classification. So, here what are the correct statements suitable for matching, that is 1 and 2, right? And 4, if you see, both A and B are acellular organisms, true. Yes, both A and B are called as acellular organisms. Viruses are acellular organisms, or they can be described as living entities, or they can be described as crystalline units. So, here 1, 2 and 4. Option D is the right answer. 1, 2 and 4 are correct and option D is the right answer. I'll tell you I'll tell you the preparation strategy at the end of this video. Just be there, hold on, we have just 2 more questions to deal with. Yes. Match column 1 and column 2. Select the correct option from the codes given below. Okay. So, see again examples based match the following only. Monera. Monera includes what? Oscillatoria and Bacillus, rod shaped bacteria. This is option 2. Protista, if you see, Euglena, euglenoids, trypanosoma, protozoans, unicellular eukaryotes, that is option 3. Fungi, if you see, Mucor and Penicillium, C, which is 4. Plantae, Chlamydomonas and Solanum. Chlamydomonas, which is the unicellular green algae, and Solanum is the potato plant. So, D is 1. Animalia, Felis and Panthera. Clear, right? It is as simple as it is. Now, you have to identify the options carefully. A is 2, B will be 3, C is 4, D is 1 and E is 5. Option B is the right answer. Next question ma. Yeah, this one, last question of today's session. Focus and listen. Read the given statements about the lichens and select the incorrect ones. So, here he is asking us to identify the incorrect statement. They represent an example of commensalism. Okay. So, the statements are about lichens. In lichens we see symbiotic association, that is mutualism, not commensalism. And then second statement, algal partner obtains water, minerals from the fungus, fungal partner obtain foods that is prepared by the algae. True. And they do not grow in polluted areas. Yes, they are very sensitive to polluted areas, especially for the sulfate pollutants. Okay. The mycobiont is a green, the mycobiont is mostly a green algae. No, mycobiont is mostly fungus. Phycobiont is green algae. The phycobiont is mostly green algae or cyanobacteria. True. And these constitute pioneer community in case of hydrosear, that is plant succession. Yes, this is also true. Option 6 is correct. So, you look into the statements properly. Which of them are incorrect? 1, 4, okay. 1, 4 are incorrect. Okay, 1 and 4. So, here it is not given, 4 and 6 as well. So, here if you see, these constitute pioneer community in mesosphere or xerosphere, not hydrosphere. Mesosphere or xerosphere, not the hydrosphere. So, option D will be the right answer. Okay. So, 1, 4 and 6 are the incorrect statements. So, this is all about the today's class. With this we have finished out the advanced questionnaire practice and NEET previous year questionnaire practice of biological classification. In our next session we are going to look into the NEET previous year questions on plant kingdom, okay, which is very important. So, do not miss that session at all. So, here in the comment section there is a question asked by a student, that is, ma'am, how to start reading NEET while studying in the 10th. So, ma, look here. So, when you are in 10th only, build up your foundation concepts, because most of the concepts are in connection with your 11th standard concepts. For that there is a playlist in the NEET World channel which is for the bridge course, where we have connected all the concepts of your 10th standard with 11th standard. Take those videos as reference and look into the concepts in detail, practice NEET objective accordingly. Okay. So, this is the first step you have to do, which will be the foundation for all the concepts which are connecting with 11th and 12th standards. Okay. So, this is the first step you have to do, beta, and then parallelly along with your classes, just focus additionally daily 1 hour or 2 hours on the concepts which are included in the NCERT 11th. This will boost up your preparation. Okay. So, this is all I can advise you at this point of your 10th standard. Okay. And with this we are ending today's questionnaire session. We will see you tomorrow with plant kingdom previous year questions. Thank you.